How's everybody doing? Everybody's quiet? Settling down? Chilling? Thinking about biochemistry? No. I'm a dreamer, aren't I? Okay. Well, I want to finish up talking about electron transport, oxidative phosphorylation. I hope I gave you an understanding yesterday of respiratory control, because with respiratory control, we can understand at the molecular level how our body functions. And that's a pretty cool thing, all right? So the last topics that I have to talk about have nothing to do with respiratory control, but they are considerations for respiratory control, okay? And you'll see why that's the case. I start by telling you something that is a limitation for cells. And the limitation for cells is that NAD and NADH cannot cross the mitochondrial inner membrane. They can't cross it. So if I generate NADH out in the cytoplasm and I want to convert it back to NAD, I have to figure out a trick to get the electrons from NADH into the inner mitochondrial membrane. Okay? Because the molecules themselves will not travel, and they have to get inside the membrane before they can dump their electrons. Okay? So to do that, cells use what's called a shuttle in order to get the electrons in, a shuttle. So a shuttle is something that carries the electrons for NADH, and then when it gets inside the mitochondrion, it dumps those electrons off onto something else. Make sense? Okay? So that's what a shuttle does. There are two main shuttles that I'll talk about. The first of those is the glycerol phosphate shuttle, and it's actually the easier of the two to understand. It's one that's very prominent in insect muscle. And in insect muscle, this shuttle works, as you can imagine, very rapidly. And for insects, having their muscles function rapidly is very, very important. Okay? Because a fly has to be able to fly away as you're trying to swat it, right? It has to be able to work instantaneously. So this shuttle works, as I said, in a fairly simple fashion. NADH has electrons, okay? It wants to get rid of those electrons, and it can't cross the mitochondrial inner membrane, so it can't dump its electrons off directly into the matrix. So what it does is it takes the electrons on NADH, and it gives them to an intermediate from glycolysis. And the intermediate from glycolysis is glycerol. Is dihydroxyacetone phosphate. Okay? When it gives its electrons and protons to DHAP, it converts it into glycerol 3-phosphate. That's what it's actually doing there. It's giving its electrons to DHAP to make glycerol 3-phosphate. Okay? You'll notice this is a ketone, and you'll notice this is an alcohol. That indicates a reduction that has occurred. Well, glycerol 3-phosphate can be moved across the mitochondrial inner membrane via a shuttle system, via a transfer system that does work. So glycerol 3-phosphate can move in, and when glycerol 3-phosphate moves in, it donates its electrons to FAD to make FADH2 and reestablishes, remakes, whatever we want to say, dihydroxyacetone phosphate. So we've completed the cycle. We started with dihydroxyacetone phosphate, made glycerol 3-phosphate, made dihydroxyacetone phosphate. Now, the only difference was we started with NADH, and we ended up with FADH2. As I mentioned yesterday, FADH2 does not pump as many protons as does NADH, which means that this process is not very efficient. It's losing some of the potential energy as a result of doing this, but it has the advantage of being very, very quick. Okay? In our cells, we have a different shuttle system that exists, and I want to show you that shuttle system. It looks a little bit more complicated, but I'm going to simplify it for you. I keep saying that, don't I? You say, what the hell are you talking about? You're not doing anything, right? Okay, so this is the malate aspartate shuttle, and on the surface it looks very complicated, but let's look at the important players here. Okay? We have the same problem. We have electrons in NADH. We can't get the NADH across the membrane, so we have to use a shuttle molecule to carry them in. Okay? Well, that's actually depicted right here. And you can see oxaloacetate is the starting point, and we have these electrons from NADH that dump electrons and protons onto oxaloacetate to make malate. This is the reversal of the reaction that's occurring in the citric acid cycle. Okay? We make malate, and malate can make it into the mitochondrion. Inside the mitochondrion, malate gets converted back to oxaloacetate, and in the process of doing that, regenerates NADH. This process is 100% efficient. It doesn't lose any energy in the process of making the molecule. Everything else that's shown on this figure is simply going back to regenerate the oxaloacetate. Okay? So it's called the malate aspartate shuttle because it involves aspartic acid. Okay? It involves oxaloacetate, involves glutamate, involves alpha-ketoglutarate, and we don't really care about all that stuff. Okay? This is simply balancing the equation. So I'm not going to ask you to draw this, but I do think that you should know what the important players are. Oxaloacetate is the electron acceptor in the cytoplasm. Malate is the molecule that moves across the membrane. And NADH and oxaloacetate are regenerated inside the mitochondrial inner membrane. So this process is slower. However, it's very efficient. Okay? We don't lose any of the energy that was in that NADH in the process of this shuttle going on. Okay, questions about that? Yes, Daria. I'm sorry, I didn't hear what you said.
It was the last point that you just made about the malate.
So malate is converted back to oxaloacetate. So that's an end point for this. And the regeneration of NADH inside the membrane. So we start with NADH out, and we end up with NADH in. Okay? Clear as mud? All right. Well, now we turn our attention to another important aspect of metabolism, and that's lipid metabolism. Lipids, you may recall, include the nonpolar molecules. They include the fats. They include the fatty acids. They include the steroids. They include the glycerol phospholipids, blah, 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 right? Fats and fatty acids are where we're going to focus most of our attention because fats and fatty acids are the highest energy storage units in our body. We store more energy in fatty acids than we store in glycogen. Okay? We have about a 24-hour supply of glycogen in our body. That's about what we have. Fats can keep us going for a longer period of time. Now, the problem with fats is that fats are not water-soluble. So the body can't do like what it does with glucose and glycogen. It breaks down glycogen, makes glucose, and dumps it in the bloodstream. Bang! It's at this target destination in seconds. Can't do that with fats because fats are not soluble in the aqueous environment of the bloodstream. So fats and fatty acids have to be packaged up so that they can travel in the bloodstream, and that takes longer. So fats and fatty acids don't work well for instantaneous energy. Okay? We will see also that the regulation of these pathways, both the breakdown of fat and the synthesis of fatty acids, is very simple. It's not overly complicated. Well, how do fatty acids get released from fats? Okay? This is how it happens, and it's actually quite simple. Okay? There are enzymes that are known as lipases that will cleave the bond between fatty acids and glycerol. All right? So what you end up with are fatty acids, and you end up with a molecule of glycerol. 3 fatty acids and 1 glycerol per fat. Glycerol can be converted into intermediates in the citric acid cycle, such as glyceraldehyde 3-phosphate, and glycerol can be made back into glucose. This is the only part of a fat that can be made into glucose. None of the fatty acid can be made into glucose, and the reason for that is because, as we will see, fatty acids break down into acetyl-CoA, and in our bodies we can't convert acetyl-CoA into glucose. So the only part of fats that can go back into glucose is glycerol. It's a pretty small part. Okay. Glycerol phospholipids, you recall, are related to the fats. Fats have 3 fatty acids, 1 on each of the carbons of glycerol. Glycerol phospholipids have 2 fatty acids, 1 on each of the first 2 carbons, and the third carbon is linked to a phosphate substance. These glycerol phospholipids are the things that are in our lipid bilayers. And we can also break down those guys. We can pull the fatty acids off of them, and we do that with enzymes called phospholipases. Okay. So we've gotten the fatty acids off, and now we would like to do something with them. Okay? We don't need to worry about that. Let's ignore that. Okay. Before I tell you about how we break down the fatty acids, I do want to point out one of the regulation things. One of the regulation things is, well, how do we break down fats? It turns out that those enzymes that are there require activation by a hormone. And the hormone that they require is something like the hormone epinephrine or glucagon. Those will stimulate this pathway, which is the same pathway that's involved in regulating glycogen breakdown. The same pathway. Now, why are cells breaking down glycogen? Why? Why is the body breaking down glycogen? It needs glucose and it needs energy, right? What's contained inside of fats? Energy. So the same pathway that stimulates the breakdown of glycogen also stimulates the breakdown of fats. When that happens, the various lipases are activated. And you can see in this figure, here's a triacylglycerol, that's a fat, being broken down, one fatty acid at a time being pulled off of the glycerol. So that we end up with glycerol and 3 individual fatty acids. Okay? So there's a hormonal control to the breakdown of fat. We can decide, hey, I'm going on a diet, I'm just going to go drop all this fat, et cetera, et cetera. We're ultimately going to have to convince our body to get this process going where we have low energy, where the body says, we've got to get some more energy here to stimulate this breakdown of fat. And again, this is a slow, relatively slow process. Well, I'm going to come back later and talk about how it is that fatty acids and cholesterol and so forth move in the body. All right? But for right now, I want to assume that the fatty acid that has gotten broken down in one part of the body has traveled to another part. I should also point out that this body stores fat in specialized cells. Fat is stored in specialized cells. Those specialized cells are called adipocytes. A-D-I-P-O-C-Y-T-E-S. Adipocytes are fat cells. People call them fat cells because they're full of fat. They store fat, the body breaks it down, and then the fatty acids travel to target tissues, where the target tissues take it up and then start the process that I'm getting ready to describe to you here. So this is going on inside of target tissues. What happens? Well, fatty acids by themselves in a cell are problematic. Anybody know why they're problematic? Speak up. They're partly hydrophobic. What else? How do we make a detergent? We make a detergent by taking fat, okay, And we treat it with sodium hydroxide, break the fatty acids off the fat, so we have sodium salts of the fatty acids. Right? You're supposed to say yes. Okay? That's how we make fats. Basically, if we have free fatty acids, we have detergents. And what do detergents do to proteins? They denature them, right? So we don't want to have too many free fatty acids floating around in a cell, right? So the cell caps that fatty acid at the polar end by putting on coenzyme A. You saw CoA put on to the end of acetic acid. Acetic acid is a 2-carbon fatty acid. These fatty acids are much longer. They're typically 16 to 20 carbons long. Acetic acid won't act like a detergent because it's not long enough. It doesn't have that nonpolar part. But a long fatty acid does have that property. So the first thing that a cell does when it gets a free fatty acid inside of it is it puts a CoA on it. And you can see in the process of putting on there that is an energy-requiring process. It's a very energy-requiring process because it starts with a fatty acid, okay? It adds CoA, but it starts with an ATP and ends up with AMP, meaning it's clipping off 2 phosphates. That's something that you haven't seen before, but that means it's very energetically requiring process. And I'll ask you what kind of an intermediate we created in the process. Activated intermediate. Excellent. Who said that? 2 points. You make an activated intermediate. Okay? That activated intermediate will be important because it has a high energy bond, right? Okay. So we've made a fatty acid linked to CoA. So what? Well, we're now getting ready to oxidize that fatty acid, but then something odd interrupts the process. Okay? Something odd interrupts the process. What interrupts the process is the fact that the cell has just blown a wad of energy on putting a CoA on there, and fatty acid oxidation occurs in the mitochondrial inner membrane, but an acyl-CoA, which is a fatty acid linked to CoA, acyl-CoA, ACYL, and acyl-CoA cannot cross the mitochondrial inner membrane. The cell has just made something that can't cross the inner membrane. Duh. So what does it do? Well, as it gets to the mitochondrion, there are enzymes near the mitochondrion that pull off the CoA and replace it with something called carnitine. All right, so here's the acyl-CoA that's out in the cytoplasm. The acyl-CoA up in the cytoplasm is right here, okay? This guy makes it across the outer membrane, but it can't cross the inner membrane. Okay? So it gets across the outer membrane. Here it is. It's an acyl-CoA that's in there. But when it hits the inner membrane, it can't cross. So in the inner membrane, in this inner membrane space, there's an enzyme that will pull off the CoA and replace it with a molecule called carnitine. The enzyme is called carnitine acyltransferase. CoA comes off. Carnitine goes on, so now we have an acyl-carnitine. And acyl-carnitine crosses the barrier just fine. Acyl-carnitine goes in. Now we're inside the matrix, okay? And then the reversal of the process occurs so that we now make acyl-CoA again. And you say, well, how'd that CoA get inside the mitochondrion? It's made inside there. So there are enzymes that will make it out in the cytoplasm, and there are enzymes that will make it inside the mitochondrion. At this point, where we have the acyl-CoA, we are ready to oxidize that fatty acid because we've gotten it into the mitochondrial inner membrane. Now we're ready for a process called beta oxidation. Beta oxidation. It looks hairy, but I will tell you, you've already learned it once. All right? You've already learned this process once, at least 3 of the steps. Okay? Here's our CoA. When we look at fatty acid oxidation, if we start with the carbonyl carbon, which is the one that's linked to the S on the CoA, if we start with the carbonyl carbon and call that either number 1 or alpha, that means that if you see in the turquoise here, everything that happens occurs between, that's alpha, beta, gamma, but in any event, everything happens between carbons 2 and 3. Everything that happens in fatty acid oxidation occurs here. Okay? So the first thing that happens in the process is an oxidation. And the oxidation involves the removal of 2 protons and 2 electrons to make an intermediate that has a trans double bond between carbons 2 and 3. Have you seen that before? Anybody know where they've seen that reaction before? It wasn't that long ago. As in day before yesterday. So what's happened is we've started with something that has this guy right here. We've pulled off 2 protons, 2 electrons, and made a double bond with a trans bond right there. FAD accepted the electrons to become FADH2. There's a hint. The answer, I'm going to tell you since you don't have it, is when we oxidized succinate in the citric acid cycle. That was the reaction that took 2 protons, 2 electrons away. It made that trans double bond that I showed you at that time, and the electrons went to FAD and made FADH2. Same reaction. That also occurred between carbons 2 and 3. Okay? The next step in the process, water comes in and adds across that double bond. And when water adds across the double bond, the hydroxyl goes on to position 3, and the hydrogen goes on to position 2 and leaves behind a single bond. That's the reaction that happened after the succinate reaction in the citric acid cycle. That was the reaction that took fumarate to malate. Exact same reaction, exact same thing happened. Okay? Fumarate to malate. The next step in the process is an oxidation. NAD is becoming NADH, meaning that the hydroxyl is becoming a ketone. And that happened in malate going to oxaloacetate. So we've just done 3 identical reactions to the reactions that were done in the citric acid cycle. 3 identical reactions. Succinate to fumarate, fumarate to malate, malate to oxaloacetate. We had the same electron carriers, nothing different. In the next step, we've actually completed beta oxidation because in the next step, there's an enzyme called thiolase, Thiolase, and yes, that's a name you should know. Thiolase breaks the bond between carbons 2 and 3 and puts a CoA onto the remainder. What does that mean? Here's our starting molecule. Here's the break-off of the 2-carbon piece. All right? That makes acetyl-CoA. And here's the fatty acid that's been shortened by 2 carbons. Now this guy, this fatty acid right here, will go back up and start the process all over again. Now one thing I didn't tell you, and it actually relates to human health, so I want to tell you something about that, is the very first enzyme in this pathway. Yes, question? Oh, is the noise? I'm sorry. Should have said something earlier. It's the other one then. Good. Yeah. Okay. All right. So there's an enzyme that's the very first enzyme in the pathway that has an important implication for human health. I need to give you the name of this enzyme, which means that you'll need to know the name of this enzyme. Okay? Okay? It's called acyl-CoA dehydrogenase. Acyl-CoA, and then the word dehydrogenase. Acyl-CoA dehydrogenase catalyzes that first step, the removal of those hydrogens and electrons from the acyl-CoA to make this trans-intermediate. And by the way, I don't care that you know the names of all these. All right? I really don't. It doesn't matter. But the acyl-CoA dehydrogenase is important, and the reason it's important is that it's been found that in many of the infants that suffer, that die from sudden infant death syndrome, they have a deficiency in that enzyme. There are 3 forms of that enzyme. One that works on long-chain fatty acids, one that works on medium-sized fatty acids, and one that works on little baby fatty acids. The one that's deficient in SIDS is the one that's in the medium-sized fatty acids. Now, it's not the case for every infant that dies of sudden infant death syndrome. So I don't mean to say it's a cause and effect. But many of the infants who have died of that have actually been deficient in that enzyme. Okay, questions about that? You guys look worn out. How many people were outside before they came here? How many people want to go outside? I'm not letting you go. I'm sorry. Maybe everybody should stretch. Why don't we stretch? Okay, get up, stretch. Breathe deep. Get that oxygen in the brain. Come on. Everybody. It's also hot in here today. Is it just me or is it hot in here? Yeah. Yeah. No, the temperature in here is weird. You know, it's just weird. When I complain about the temperature, I think they've actually made it warmer. And that's what, on a hot day like today, you notice. So. Okay. Now I'm going to show you one more thing on this diagram, and then we'll move on. Okay? The one thing I'll show you in this diagram is right here, that hydroxyl. You'll notice that hydroxyl is in the L configuration. That fatty acid form is in the L configuration. The reason I show you that is when we go to do fatty acid synthesis, we'll make an almost identical structure, except for it's going to be in the D. That's one way you can tell a fatty acid that's being oxidized from a fatty acid that's being made. L versus D. Okay, questions about this? Yeah, this is the oxidation pathway we're looking at here. Yeah. Okay. Okay. Let's turn our attention to the breakdown of a long-chain fatty acid. Okay. This is an 18-carbon fatty acid. And that process that I just showed you will occur on this fatty acid 8 times. The last one splits a 4-carbon piece in 2. It doesn't occur 9 times. It occurs 8 times. Okay? But that same process occurs here, 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 here. And when we're done with that, we will get 9 molecules of acetyl-CoA. What we're going to see is that fatty acids are built, and they grow by 2 carbons at a time. They start with a 3-carbon molecule, but they grow by 2 carbons at a time. And that's the next thing I'm going to, well, actually, let me see. I think it may be one of the things I'm talking about before that. Okay. Oh, yeah, that's right. Okay. Let's talk about odd-chain fatty acids. Odd-chain fatty acids. All right. What is an odd-chain fatty acid? Well, most fatty acids in the body have an even number of carbons. 24, 22, 20, 18, 16, et cetera, et cetera. And that works out really well when you go breaking them down, because when you break them down, you get down to that final 4-carbon piece, you split it in half, and you get 2 2-carbon pieces. Well, what if you eat some food that has 17 carbons instead of 18? what's going to happen when you get down to the end? When you're going to get down to the end, you're going to have a 3-carbon piece, and the cell will not cut that 3-carbon piece any further. So it's got to handle it in a different way. Okay? So odd-chain fatty acids, when you get down to that last 3-carbon piece, it's got to do something with them. That 3-carbon piece is called propionyl-CoA. P-R-O-P-I-O-N-Y-L. There you go, propionyl-CoA. Okay? Well, how does a cell deal with that? Well, it turns out that it's actually kind of an odd set of reactions that the cell goes through to do it. In this schematic, I simplified it quite a bit. Okay? Let me tell you what happens. First, the cell decides that, well, if we have 4 carbons, we can make it into something that's useful. So the cell adds a carbon by adding a carboxyl group. Okay? And it puts that carboxyl group not on the end carbon, but on the middle carbon. Don't ask me why the cell does that. Okay? Not only does the cell put the carbon on the middle carbon, it puts it in the wrong configuration. D versus L. What's the wrong configuration? Well, it means it's got to move it into the other configuration before it moves it. So the cell is really doing something stupid. It's making something that it has to rearrange before it can rearrange it again. 3 steps instead of 1 step. If it puts the carboxyl group on the end, we would end up with the product, which is succinyl-CoA. But it doesn't. It starts with propionyl. It goes through 2 gyrations. You only see one of them on here. And finally ends up with succinyl-CoA. So it ends up with what it wants to have, but it takes a long, roundabout way to get there. Okay? Succinyl-CoA, of course, is an intermediate in the citric acid cycle, and that's how it's metabolized. So that then enters the citric acid cycle, and everybody's fine. So that's how odd carbon fatty acids are oxidized in the cell. You see these various other things, some amino acids, valine, etc. These are molecules that give rise to these different intermediates that you can see here. Okay? Okay. Well, here's something you're not going to like. So we might as well just get it over with, right? Oxidation of unsaturated fatty acids. Okay? Unsaturated fatty acids. Everything's simple in beta oxidation. You got 4 steps. Dehydrogenation, addition of water, oxidation, and break off of that 2-carbon piece, thiolase reaction. Very simple. Well, what happens if you have a double bond occurring in that process? Oh, by the way, I should point out something here, and I mentioned it, but I didn't emphasize it. All right? Look at that intermediate. What configuration is that in? That's a trans bond, right? Is that a trans fatty acid? It's actually not. Okay? We generate trans bonds in the process of oxidation. We also generate them in the process of synthesis, right? And this has nothing to do with there being a trans fat or fatty acid. And you'll see why that's the case as we talk about it here. Right? What if I have unsaturated fatty acids? As I've said earlier in the class, almost all of the naturally occurring double bonds in fatty acids are in the cis configuration. Okay? How do we get trans fatty acids? Well, we get trans fatty acids by hydrogenation of vegetable oil. That's done to change its properties and also to lower its melting point. Okay? That generates trans bonds, not in a CoA-containing molecule, but in a free fatty acid. And the position of that trans is completely variable. The position of the trans that you saw in what I've showed you is occurring at a very specific place between carbons 2 and 3. Okay? We don't know why trans fatty acids are problems, but we do know that they are problems. Okay? You see increases in inflammation. You see what people believe to be increases in bad cholesterol, LDLs. Okay? We see bad things associated with trans fatty acids, but we don't exactly know why that's the case. All right. How do we oxidize an unsaturated fatty acid? Well, an unsaturated fatty acid coming from a biological source is largely going to be in the cis configuration. Let's imagine we have something here like this fatty acid, acyl-CoA. It's from oleic acid. And this fatty acid has a series of carbons. You can see there's 9 carbons before we get to this one. So we're going to have to oxidize these guys 2 carbons at a time to break off pieces to get finally down to a structure that we've got to deal with. So here it is going on right here. We see oleic-CoA going through 3 cycles of beta oxidation, meaning it's splitting off 6 carbons. And that gets us into the configuration you see right here. In the configuration you see right here, you see a trans double bond between carbons 1, 2, 3, and 4. I'm sorry, I say trans, a cis double bond between carbons 3 and 4. A cis double bond between carbons 3 and 4. In beta oxidation, where do you want the bond to be? Between 2 and 3. And what configuration do you want it to be in? Trans, right? In beta oxidation, it's in the trans. Well, it turns out it's very simple. There's an enzyme called enoyl-CoA isomerase that will convert a cis 3,4 into a trans 2,3. That's simple. Okay? Then fatty acid beta oxidation continues just like before. Okay? So we added one enzyme, and that one enzyme converted a cis 3,4 to a trans 2,3, and then everything else was just beta oxidation, just like before. Everybody with me? Well, that's really nice if the cis bond is between carbons 3 and 4, but what if it's somewhere else? Well, let's see what happens when it's somewhere else. Here's oxidation of linoleic acid. It's got a bond in the same place, the first bond in the same place that oleic acid had. Meaning that we can go through 3 cycles of beta oxidation and break off 6 carbons, and we end up with a cis bond between carbons 3 and 4. Everybody with me? Enoyl-CoA isomerase comes in, converts a 3,4 cis to a 2,3 trans. Beta oxidation continues to break off those 2 carbon pieces, right? Right. And that leaves us with this structure. And this structure has 1, 2, 3, 4, 5. It has a cis between 4 and 5. Okay? What does the cell do with that? Well, it turns out it won't treat that as a substrate for beta oxidation. Okay? It starts the process. It makes a trans between 2 and 3, but it can't go any further. This was the first step in beta oxidation. It pulls those hydrogens off, but the next enzyme won't touch it. We've got 2 double bonds. Cell throws its hands up in the air. What am I going to do? Okay? Well, fortunately, it has an enzyme that I'm not going to hold you responsible for knowing, so I'm not going to repeat its name. And that enzyme converts those 2 bonds in that configuration into a cis 3,4. So we converted 2 bonds that are double. That takes electrons. The electrons come from NADH into a cis 3,4. How do we deal with a cis 3,4? Okay? So we've got enoyl-CoA isomerase that converts a cis 3,4 into a trans 2,3, and everybody's done. So even though this looks complicated, there were only 2 enzymes that were necessary to oxidize the multiple double bonds that are in this molecule. An enzyme whose name I didn't give you, and enoyl-CoA isomerase. Everybody understand that? Now, I'm not going to ask you to draw all this out. I want you to have a general understanding of what's going on here. And I think you should specifically be able to tell me what kind of a reaction enoyl-CoA isomerase catalyzes. It catalyzes the conversion of a cis bond between carbons 2, 3 in a fatty acyl-CoA into between 3 and 4 in a fatty acyl-CoA into a trans 2, 3. That's what it does. And that enables all these processes described to occur. That oxygen didn't do anybody any good. Yes, question. This is linoleic. Gesundheit. Allergic to biochemistry? Okay. Any other questions? All right, I'll tell you what we'll do. Let's, ketone body. I'll tell you what, let's do ketone body metabolism. And then maybe an extra credit question. Or is that a no? An extra credit question. Then I've got a song for you, okay? Ketone bodies. Let's talk about ketone bodies. Why do I talk about ketone? How many people here have heard of ketone bodies before? Okay. Mostly nutritionists, okay? So we talk about ketone metabolism and so forth. What are ketone bodies? Ketone bodies are very interesting molecules. And they're interesting molecules because they're a sort of escape hatch for your body. Remember that your body cannot cannot synthesize glucose from acetyl-CoA. So if you were to stop eating all carbs, that is, if you were on a starvation diet, you were not eating anything, you would not be eating any carbs, and I said you've got about a 24-hour supply of glycogen and glucose in your body. Your body needs glucose, your eyes need glucose, your brain needs glucose. So, and they can't make it, they're reliant on the other tissues of the body to provide it for them. What happens? Well, one thing that happens is you can convert fatty acids into glucose. So some of your protein will get broken down, especially if you're on a starvation diet. Proteins break down to amino acids go into glucose. A more immediate way of dealing with that is for your body to break down fatty acids into acetyl-CoA and make ketone bodies. Now, ketone bodies won't get made into glucose. So they're not well, in a sense, they're substituting energy-wise for glucose, but they won't make glucose. But they can be used by the brain and by the eyes, which is very important to keep going when you're not eating. So what happens? Here's two acetyl-CoA molecules. Two acetyl-CoA molecules can be converted into a 4-carbon, that is, they're joined together to make a molecule called acetyl-CoA. 4 carbons, 2 carbons to 4 carbons. We add another acetyl-CoA, and now we've got 6 carbons. We're getting to the ketone bodies. This 6-carbon molecule is called HMG-CoA, and it's a really important molecule because if it doesn't go to making ketone bodies, it goes to making cholesterol. We'll talk about that later. But if it's making ketone bodies, an acetyl-CoA is split off, and a 4-carbon molecule called acetoacetate, that's the first name I want you to know up there, acetoacetate, is made. That's one of the ketone bodies. Acetoacetate can be enzymatically converted into another molecule we call a ketone body, although it's not a very good one. It's not even a ketone, but it's called beta-hydroxybutyrate. That molecule is fairly stable, and it will travel in the bloodstream from the place it's being made, say in your liver, to your brain. When it gets to your brain reverses all those reactions and releases 2-acetyl-CoAs, which can be used in the citric acid cycle. So the body has found a way to get energy to the brain when it needs it when you're not eating glucose. What about this thing over here? Well, acetoacetate itself is chemically unstable. And it will spontaneously decarboxylate, losing a carbon dioxide. And when it does that, it creates acetone. Acetone is an organic solvent. You can smell it in fingernail polish remover. Okay? You probably know what that smells like. And you can actually smell acetone on the breath of a person who's making ketone bodies. If you ever smell acetone on the breath of somebody who's on a normal diet, they're not starving themselves to death, you want to tell them to get to the doctor, because that's oftentimes a sign of diabetes for somebody who doesn't know it. With diabetes, blood glucose levels are going way up, way down, way up, way down, and when they go way down, the body's got to make ketone bodies to keep things going, and people aren't aware of it until somebody tells them, you smell like acetone. Okay? So that's an indicator of the fact that a person might have a form of diabetes if that's happening. Questions about that? Okay, we're going to do a quick song, and then we're going to do extra credit. So here goes the quick song. It's called, I'm sorry, I got the wrong one. It's called Fatty Acid in Our Cell. What did I just do? Yeah. Okay. No, that's the wrong one. What did I do? Oh, blast it. Hold on. Here we go. All right. This will help you with fatty acid oxidation. That's not it. Ah. I know. Ah. There we go. No? All right, we'll have it tomorrow. How about we do the extra credit? Okay. We'll do 2 songs tomorrow. All right.